Hi everyone, I am back with yet another new chapter, Plant Cell Culture and Applications. This video is the part 1 which covers the initial introduction of plant tissue culture, which covers what is totipotency, history of plant tissue culture, medium and components of the medium of plant tissue culture. What is plant tissue culture? The aseptic culture of plant protoplasts, cells, tissues, or organs which lead to cell multiplication or regeneration of organs or whole plants. So there are two key terms here. One, aseptic culture. Here we are doing in vitro culture, means we are culturing it in laboratory conditions. Aseptically means it is preventing we are preventing any sort of contaminations from pathogens. Okay, and second, the material, starting material, it can be plant protoplasts, cells, tissues, or organs. So the starting material, we call it as explants. So what happens here, we can either have cell multiplication or we can have regeneration of the organs or the whole plants by the formation of the roots or shoots. Plant tissue culture often produce clones of plants which are true to type of the selected genotype. So in plant tissue culture, whatever products you are getting, like when you start with an explant, okay, the seedlings that arise from that, they are all clones. They all are identical to each other, and it is all true to type of the selected starting material. The control conditions provide the culture and environment conducive for their growth and multiplication. This is very important. Unless the conditions are not controlled, you cannot have a proper plant tissue culture being implemented in the laboratory. These conditions include proper supply of nutrients, pH, which is very important, adequate temperature, and proper gaseous and liquid environment. So we need to have proper control conditions for the plant tissue culture. History of plant cell culture. Gottlieb Haberland, a German physiologist, is regarded as the father of plant cell culture. He first isolated single palisade cells from the leaves in not salt solution enriched with sucrose in 1902. Next, very important question. Why does tissue culture work? It's because of the ability of totipotency. What is totipotency? The ability of a cell to differentiate and develop into a whole plant when given the correct conditions. We were looking at the conditions like correct temperature, correct humidity, correct photoperiodism, correct nutrient, correct pH, all the conditions should be appropriate. Then every cell can be differentiated and develop into a whole new plant. This is because every cell has the genetic potential of the parent plant, and this ability is what we call as totipotency. So what is required? First, appropriate tissue. Why appropriate tissue? Sometimes some tissues culture better than others. So we need to first determine what tissue or what is our starting material. A suitable growth medium, that is very important. And this growth medium should have energy sources and inorganic salts to supply the cell growth needs. This can be either in the liquid form or semi-solid form. Third, we need aseptic or sterile conditions. Why? Because microorganisms can grow much more quickly than plant and animal tissue, and can even overrun a culture. So whatever nutrient we are providing, it will be consumed by the microorganisms. So it is very important to maintain the aseptic or sterile conditions. Next, what is required is growth regulators. In plants, we need both auxins and cytokinins, which are plant growth hormones. Then we need frequent subculturing to ensure adequate nutrition and to avoid the buildup of waste metabolites. If you do not subculture frequently, the nutrition get exhausted and even the waste products will be accumulated. What is subculture? After a period of time, it becomes necessary due to nutrient depletion and medium drying to transfer the organs and tissues to fresh media. So, this transferring to fresh media in a periodic time in order to prevent the nutrient depletion and medium drying is what we call as subculture. In general, callus cultures are subcultured every 4 to 6 weeks. And theoretically, plant cell and tissue cultures may be maintained indefinitely by serial subculturing. Coming to what is callus, callus is nothing but a transparent, mass of cells which is obtained by continuous division of the cell material. Tissue culture laboratory. We need certain rooms to have a proper tissue cultural laboratory. So let's see what all rooms do we need. First, we need a general laboratory and a media preparation room. Next, we need transfer area where we transfer the cultures aseptically. Laminar flow hood will be a part of this transfer area. Next, we need culturing facilities or growth room. This room is very important because after subculturing or after transfer of these cultures aseptically, we keep it in this room where the parameters are in control conditions. We will have fluorescent tubes in order to maintain the light, and we will have regulators to maintain the photoperiodism. Humidity will be maintained. So this growth room is very important. Washing and storage rooms and greenhouse and containment room. So how do we process the tissue culture? First, you will isolate the cells. We use appropriate enzymes to isolate the cells, because if you're taking a tissue, we need to separate out the cells from the tissue. Now, we apply the isolated cells onto an appropriate growth medium in a culture dish or a petri plate. Culture cells by placing the culture dish in a cell incubator where a proper temperature will be maintained. Verify the cultured cells are of the cell type of interest, and then further it can be manipulated or modified for experimental procedures. Sometimes subculture cells to obtain pure culture or to bypass some problems such as senescence. So, what are the sterilization methods we use in tissue culture laboratory? The materials, that is vessels, instruments, medium, plant material, everything that we use in culture work should be freed from microbes. So this is achieved by one of the following approaches. First one, dry heat treatment, where the vessels etc. we can keep it in hot air oven where the temperature can be around 120 degrees Celsius. So that will be dry heat. Second will be flame sterilization. So that is also very required, and we use flames even in the tissue culture hood. Autoclaving, the medium as well as the spatula, forceps, knives, etc. Whatever we are using in the transfer of the cultures, these will be autoclaved. Autoclave is wet sterilization where the temperature will be around 120 degrees Celsius. 15 psi for 15 to 20 minutes. Filter sterilization. If you are using antibiotics, growth hormones, etc., if you autoclave it, it will get denatured. So in these cases, we do filter sterilization. Wiping with 70% ethanol, which is very important. This is a part of surface sterilization. The place where we are working, or the tissue culture hood, all these will be wiped by 70% ethanol. Surface sterilization, this is of the explants. So whatever plant material we are using, it is very important to surface sterilize it with sodium hypochlorite or potassium hypochlorite so that it is completely free from the microbes. So these are the various sterilization techniques that are adapted in the tissue culture laboratory. Now coming to the culture medium, what are the components of the culture medium? It will have macro elements, it will have micronutrients, it will have plant growth regulators, it will have vitamins, sugar and support system. So macro elements means these elements are present in a larger quantity, whereas in micronutrients it is present in very small quantities. So in macro elements we can see we need potassium, phosphorus, calcium, magnesium and sulfur. Whereas in micronutrients we have iron, manganese, zinc, boron, copper, molybdenum and cobalt. So all these micronutrients are part of some enzymes. So it is very important that they are present for the active functioning of these enzymes. In plant growth regulators, we have auxins and cytokinins. Auxins are very important for root formation, and cytokinins are very important for shoot formation and even for the callus formation. In vitamins, we have thiamine, which is vitamin B1. We have nicotinic acid. We have pyridoxine and myoinositol. Sugar is a very important component because it provides energy. Usually, sucrose is used. And in the support system, we have agar, which is a solidifying agent, which is obtained from Gelidium corneum, which is a red algae. We can also use agarose or sometimes phytogel. So once more, in a nutshell, the components of the plant culture medium include inorganic salts and micronutrients, vitamins, carbon source, which is around 2 to 4% sucrose, growth regulators, NAA and 2,4-D, naphthalene acetic acid and 2,4-D. Organic supplements can also be used, which include protein hydrolysate, yeast extract, malt, coconut milk, etc. Sometimes even activated charcoal is used. Plant growth regulators, two major hormones, auxins which stimulate root development and cytokinin which stimulate shoot development. When you have increased auxin and less cytokinin, it provides root development. And increased cytokinin and less auxin will promote shoot development. And cytokinin and auxin levels are same, it stimulates the. So thank you so much for watching this video. The rest of the topics will be covered in the coming parts. Thank you very much.